Risk of neurological degeneration from COVID spike protein. Neurological degeneration is a complex and multifaceted field of study, with various factors contributing to the development of devastating diseases, such as amyotrophic lateral sclerosis (ALS). Recent research has shed light on the potential role of spike proteins in damaging the FUS gene and mutating the TDP43 protein, leading to the onset of ALS. This article will delve into the current understanding of this relationship, exploring the mechanisms by which spike proteins may contribute to neurological degeneration and the implications for our understanding of ALS. ALS is a progressive and fatal neurodegenerative disease, characterized by the loss of motor neurons, leading to muscle weakness, paralysis, and eventually respiratory failure. The disease is often associated with mutations in specific genes, including the FUS gene and the TDP43 gene. The FUS gene provides instructions for making a protein called fused in sarcoma FUS, which plays a critical role in the regulation of gene expression and the maintenance of genomic stability. Similarly, the TDP43 gene encodes a protein called transactive response DNA binding protein 43, TDP43, which is involved in various cellular processes, including transcriptional regulation and RNA processing. Research has shown that mutations in the FUS gene can lead to the development of ALS, with studies suggesting that these mutations can cause the FUS protein to misfold and aggregate, leading to cellular toxicity. Similarly, mutations in the TDP43 gene have been linked to the development of ALS, with TDP43 protein aggregates often found in the brains of individuals with the disease. Recent studies have suggested that spike proteins, which are typically associated with viral infections, may play a role in the development of ALS by damaging the FUS gene and mutating the TDP43 protein. Spike proteins are a type of protein found on the surface of certain viruses, including coronaviruses. These proteins are responsible for facilitating viral entry into host cells, where they can replicate and cause infection. However, research has suggested that spike proteins may also have a role in neurological degeneration, with studies showing that they can cause damage to the FUS gene and lead to the mutation of the TDP43 protein. This damage can lead to the misfolding and aggregation of the FUS and TDP43 proteins, resulting in cellular toxicity and the development of ALS. The exact mechanisms by which spike proteins damage the FUS gene and mutate the TDP43 protein are not yet fully understood. However, research has suggested that the process may involve the activation of certain cellular pathways, including the unfolded protein response and the NF-kappa B signaling pathway. The UPR is a cellular response to endoplasmic reticulum stress, which can be triggered by the presence of misfolded proteins. Activation of the UPR can lead to the upregulation of various genes involved in protein folding and degradation, including those involved in the regulation of the FUS and TDP43 proteins. In conclusion, the relationship between spike proteins, the FUS gene, and the TDP43 protein is complex and multifaceted. While the exact mechanisms by which spike proteins contribute to neurological degeneration and the development of ALS are not yet fully understood, research has shed light on the potential role of these proteins in damaging the FUS gene and mutating the TDP43 protein. Further study is needed to fully elucidate the relationship between spike proteins and ALS, but the current evidence suggests that this may be an important area of research in the pursuit of effective treatments for this devastating disease.